Namaste children. Today in this lecture we discussed about the recombinant DNA technology process. In the previous lecture we discussed about the restriction enzymes. What is the role of the restriction enzyme? Restriction enzymes, they produce internal cuts as well as they produce cut at the ends of the DNA sequence, and they produce these cuts at the specific point, at the specific region. Okay, and these restriction enzymes, they are of two categories. They come under two categories. One, that is endonucleases, and second, that is exonucleases. Endonucleases, which produces the internal cuts, and exonucleases, which produce at the ends of the sequence. And when they produce the cut, then after that the sticky ends that are produced in the DNA, and these sticky ends, then they contain the complementary base sequence, and in this manner the DNA fragments of two regions of two DNA, they will join with the help of the sticky ends that are produced by the restriction enzymes. Now let's discuss in detail about this process, recombinant DNA technology. So first we have to take the foreign DNA that is isolated, that is extracted from the cell, and this foreign DNA, it should contain the desired gene which has to produce its own copies. Okay. Now in this foreign DNA, with the help of the restriction enzyme, cuts that are produced. Okay, so that we will be able to get the desired gene. And here you can see that the plasmid DNA or the vector DNA that we will take, and with the help of the restriction enzyme, the cut that is produced in the plasmid DNA. What is plasmid DNA? That is an extra chromosomal DNA. Okay. Now in both the cases, the same kind of the restriction enzyme that is used. Suppose here we are using the EcoR1 enzyme. If suppose here we have used the EcoR1 enzyme to produce the cut, then here also we will use the same enzyme to produce the cut. In the previous lecture we discussed that how the EcoR1 it functions, that how it produces the sticky ends, the overhanging structures. Okay, so when restriction enzyme it produce the cut here, then the sticky ends that are produced, and here also the sticky ends that are produced by the action of the restriction enzyme. Okay, now this sequence, it will be joined with the plasmid DNA by the help of the ligase enzyme. Okay, by the help of the ligase enzyme, this sequence it will be joined here, and then it will form the recombinant DNA. This is known as recombinant DNA. And where this gene, the desired gene that is linked in the plasmid DNA, where the origin of DNA replication that is present. Okay, because by its own this gene that is not able to replicate. It is able to replicate only in the presence of the origin of replication region, and so that's why it is joined, it is linked with the origin of replication of the chromosomal DNA. Now rDNA that is formed. Now what will be the next step here? The next step, that will be the transformation process. Transformation process means that when we will transfer this, what? rDNA, that is recombinant DNA, into the cells. Okay, into the host cells for producing the more number of copies. So this process is known as transformation. Okay, so when transformation will be there, then what happen? This plasmid DNA having the desired gene, it will produce its own copies. Okay, the copy number it will be increased then. So in this manner the recombinant DNA it works. The recombinant DNA technology it works.